Hi, my name is Hannah Phillip.
And I'm Sami Martin. And we're in period 6 biotechnology. And this semester we've been working on an experiment in which
It's called the Silencing Genome Lab, and what we've been doing is learning about RNAi and how RNAi can turn off genes, and therefore by turning off the gene we can find the function of the gene in C. elegans.
So some methods we used were chunking, which is where we cut a chunk of the agar from the starter plate and put it onto the feeder plate. And the starter plate has NGM light, but the feeder plate has NGM light and IPTG agar, which they feed off of and will hopefully silence their genome. So there are 6 stages that C. elegans experience through their lifetimes. And here's an example of an egg. And this is an L1, L2, L3, L4, and adult.
As the C. elegans grow with age, they are getting longer, but then also they get fatter. The C. elegans like to clump up in little, like spaghetti-like formations, and the movement is very S-shaped like.
After a couple of days of observing our C. elegans and letting them continue feeding off of the bacteria, we noticed a few changes in the phenotype. First being that they were moving much slower and not in an S-like fashion. Our worms are actually like spazzing. The change of this phenotype can be observed in the L3 and L4 levels.
At first, we believed that the twitching and jerky movements of the C. elegans could be either related to the neuron science of the C. elegans, or the muscle movements, because we knew that like something in the brain could affect the way the C. elegans moved. But after researching the function of UNC, we realized it was an enzyme produced in the muscle. We had gene A is actually UNC22, so UNC22. And this gene creates a protein called twitchin, and twitchin is responsible for smooth muscle contraction. To trigger the RNA pathway, you need to have dsRNA that enters the cell. We did that by feeding our wild-type C. elegans to the bacteria containing dsRNA on the starter plate.
The next step of this process is when the dicer goes in and chops up the dsRNA into smaller pieces called siRNAs.
So then the siRNA binds to proteins, forming RNA-induced silencing complex, which is also known as RISC.
So the siRNAs are double-stranded, and so then dsRNA chunks them off, leaving them single-stranded.
RISC goes around probing the cells for mRNA that it recognizes, so the one that it's carrying.
Okay, so a final step of this process is when the RISC chops up the mRNA, leaving it unable to be translated into a protein. So we gained a lot out of this experience. We actually saw the RNAi process happening instead of it just being the COP analogy. And we had some trouble starting this, but after we got it to work,
it was cool to see what the function of a gene was without actually being told. We learned it by ourselves by actually turning it off. So like kind of reverse psychology, I guess, but it worked out really cool, and we got to learn the function of the RNAi, the gene, and we learned more about C. elegans, which is something that we could use later on in our biotech life.